Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, I have a few announcements to make. We are launching our GPCR consulting services. More information will be available soon. In the meantime, if you need support with your R&D project, understanding the pharmacology of your receptor, or need help getting an idea off the ground, email us at hello@drgpcr.com. We have fantastic talks coming up at the Dr. GPCR Virtual Cafe. Visit drgpcr.com/virtual-cafe to see our upcoming live events. Get your tickets today on Eventbrite. Don't forget, space is limited. To become a speaker or to sponsor an event, you can fill out the form on the main page of the Virtual Cafe. Go to drgpcr.com/virtual-cafe today. Are you subscribed to our monthly newsletter? Stay on top of all your GPCR news by subscribing today at drgpcr.com/newsletter. Also, to watch previous episodes of the Dr. GPCR Virtual Cafe or the podcast episodes, subscribe to our YouTube channel today. Last but not least, stay tuned for more information about the upcoming Dr. GPCR Summit held between September 13th and 19th, 2021. Visit drgpcr.com/summit for more information and how to submit your talk. And now, let's dive in. Hello, everyone. This is Yamina from Dr. GPCR. I'm super excited today to talk to Dr. Sam Hoare for PharmaChanics. Hi, Sam.
Hi, Yamina. Glad to be joining you today. Thanks for having me.
Thank you, Sam. Thank you for taking some time on your Saturday morning to come and talk to us.
Yeah, I'm really. I've really appreciated the podcast. You know, this year's been difficult. Haven't been able to get to conferences and hear scientists. So having this every week, just go for a quick drive in the car and listen to science for an hour has been really soothing. So really grateful for your efforts here.
Thank you so much. Thank you. I really appreciate it. And it's been soothing for me too, as well, to talk to so many scientists and so many fantastic people. So we're going to start with the first basic question is, I'm sure a lot of people in the GPCR field know who you are. You participated in the Dr. GPCR Summit, and you're really active on Twitter. But can you tell us a bit about yourself, please?
Sure. So I guess I'm really at heart a pharmacology data analyst, and GPCRs are for me the most fun system to work on in pharmacology. So that's kind of why I'm in GPCRs. But I really enjoy analyzing data, helping other people to analyze data, and to invent new methods for data analysis that help us get more insight out of the data we're generating. And I've done this in sort of multiple roles in the traditional PhD and postdoc. And then also I spent 15 years in industry working directly with medicinal chemists in making compounds. That was a lot of problem solving. And then in the last few years, I've decided to be a consultant and apply the data analysis and sort of what I've learned over the years to help other people solve their problems. I really enjoy the problem solving and the data analysis. And I'm grateful to be working in a world where I can make a career out of it. It's really wonderful. So
that's fantastic. And we'll get back to the consulting part, because I think that is something that is a field or a vocation that not everyone in the GPCR field, or at least not only the GPCR field, but as scientists know that it's a career path that can be rewarding and difficult, but at the same time as rewarding as any other career path that you may choose, as long as you enjoy it. But let's take a step back. When did you get interested in science?
That's a great question. I was talking to my cousin, and she's like, if you're doing this podcast, you've got to imagine you're talking to your 5-year-old nieces and nephews. So I think for me, it really started when I was in college, and I was always interested in science. I picked up a biology textbook when I was about 14 and just was really drawn to it. And I think in college, I kind of had a couple of spare summers, and I worked in the lab of one of the professors there who was really into cellular signaling. And this was back in the 80s when we a lot of this stuff was just being worked out. So it was an exciting time. And I remember he was, one time I was working in his lab and he came in and he was really excited and he showed me this calculation he'd done. And all he'd done was taken two measurements. He'd measured oxygen consumption and heat output. And he was able to calculate from that the number of molecules of ATP that the cell was making every second. And I was like, wow, that's cool. You can apply math to basic measurements and you can get some profound insight out of it just by knowing how to analyze the data. And for me, that was really something. And so that's really how I got turned on to science. Also, when I was when I did my undergrad, I did a year out in industry, which was the pharmaceutical industry at the time. And that I found really exciting. The idea that we can invent new medicines just using the ideas in our head was really something. And I really liked the dynamism of the industry, people working together as teams. And the whole experience was just kind of thrilling. And I wanted to be a part of it once I'd had a taste,
This was in the UK, correct?
Yeah, this was back in the UK. Yeah.
So after your college, then your undergrad, how did you decide to pursue a PhD? Was it, the fact that you realized that you can work in a team in a pharmaceutical context, but also that you can actually measure something physiologically and you can try and understand it using math?
Yeah, I think at the time, at the time I didn't really know my motivations, right? And I thought once I'd finished college, I'd go and work in the industry. And I was thinking about maybe doing a pharmacy degree. And my decision was kind of made for me. And right at that time, the industry was going through a tight patch. And so I thought, oh, well, maybe I'll just go do a PhD. And it turned out that was the right career option. I'm kind of blessed in that way. And yeah, and so for my PhD, I went to work in the lab of Philip Strange, who is somebody who the old school British pharmacology mafia will know very well. And I got from there just a really rigorous pharmacology education. But Philip was the postdoc was a postdoc for Daniel Koshland, who wrote the textbook on allosteric modulation. And so the project I worked on in Philip's lab was actually allosteric modulation of receptors, dopamine receptors, GPCRs. That was my first encounter with a GPCR. It was just pure coincidence. And what I found really, the real thrill of working in that field was I realized that we could understand something about how the protein was functioning by measuring its activity. This was in the days before we could get structures of GPCRs, and we could work out that a drug was allosterically modulating the receptor by designing the right experiments and then by applying math to the data. And at that time, a postdoc in the lab was David Hall, who's sort of classical pharmacological theorist now, works for GSK. And I'm really grateful to him. He took the time to sort of educate me on how to do the receptor theory math. And Philip helped out a lot too for the bigger picture. And at that point, I was really hooked on applying math to pharmacology data to understand how the ligand is working. And that was a lot of fun. And there was an aha moment in there as well that you often talk about, where I'd figured out in my head that the way this protein was working is that it had an allosteric mechanism and also a competitive mechanism.
Yep.
And to prove that, I had to derive the math to describe that, and then analyze the data. And when I analyzed the data, and it was in the very first version of GraphPad Prism, the curve went right through the middle of every data point, and that that's probably the high I've been chasing ever since. That was that was really something.
It's so interesting that you, quote unquote, almost accidentally decided to do a PhD, and it ended up in a context where it took you back to your undergrad experience, where you were able to apply math to understand a physiological response.
Yeah, God works in mysterious ways, as we say. So, but it was,
Agreed.
yeah, I think I like listening to your podcast because it's like you hear your own story, and most people, when they're early in their careers, they're just kind of stumbling along and following their nose, and
Yeah.
it's amazing. And I like your story in that regard. You just kind of you've had the courage to follow your convictions in what you want to do with your career. I think that's important.
Thank you. And I think one thing, and at the time I thought it was a naive way of looking at it, but now thinking back, I think it was more about having fun. It was more tackling questions. So I did a PhD and a master's because I was having fun in the lab. I was very much enjoying working, preparing experiments, doing the experiments, but also analyzing the data and coming up with answers to hypotheses, or at least, some kind of answers.
Yeah, I've been browsing some of your papers, obviously, since we're starting to collaborate a bit, and that's what comes through in your papers. It's like, I'm having fun doing this.
I had a blast.
Yeah.
Even on difficult moments. So the 2016 paper on CXCR3 alternative variants was, I think, the culmination of I'm having fun, but these receptors frustrate me, and I am going to win. And it was such it was so interesting in the sense that, so you have these 3 splice variants of this chemokine receptor, and one has a longer N terminus, the other one is much shorter. So you think that it has only 5 transmembrane domains. And when you're trying to measure their activity and you do the experiment 8 times and only 1 out of the 3 does something, and then you're wondering, is it me? Is it the cells? Is it the compounds? What is going on? But when you do it the 8th time, you're like, yeah, no, it's not me. This is it. This is it. So it was kind of a frustrating and then fun thing to do as well. And I think the most interesting part of this story was when I was measuring cyclic AMP, is that with the B variant that has, I think, 100 much longer amino acid N-terminal tail, and it wasn't doing anything with any concentration of CXCL11 unless you shot at it with 100 nanomolars.
Yeah.
And then I thought, oh, it may be just, the fact that it's just too much ligand, or it's not specific, or it's lacking something. But then you could inhibit that response with PTX.
Yeah. Yeah.
Which suggested that there was something going on. I just didn't have the right combination of proteins in the cells. But still, I think it was a fun time.
Yeah, this is science in action, isn't it? Not always easy.
Yes, it was. But then, so you during your PhD, you were enjoying, you learned pharmacology, you were interested in allosteric modulation of GPCRs. Were you also performing the experiments along with data analysis?
Oh, yeah, very much so. Yeah. So, and I was enjoying the experiments, but I'm not a great technologist. So I've had the pleasure of working with people in industry who are just genius experimenters who can get anything working. And so, during my PhD and during my postdoc, I was doing a lot of experiments at the bench and was learned a tremendous amount from people who took the time to teach me. But but I was always, my talent really is in the data analysis. So people would always come to me for that rather than for doing the experiments. And one of the reasons I sort of left and set up my consultancy really was because biology was becoming so technical, and which is a good thing. The new technologies we have now, we can investigate pretty much any question, but that was never my strong suit. So it was like I decided to specialize in the data analysis. And that was that. But I think for GPCRs, I think you're often asking why GPCRs, and I think it gets back to what you just said, which is it's good fun. You know, I speak as somebody who's not a great technologist, but I was able to do loads of experiments because they're fairly easy to work with. We can do binding assays to interrogate how the ligand is binding. We can do functional assays. We can measure second messengers, and all of these are fairly straightforward assays. Even back in the day when we were using the column method to measure cyclic AMP, which we all like to laugh about, it was generally pretty straightforward. Contrast that with trying to measure the activity of an ion channel in an electrophysiology rig, and it's just it's so easy to work with the GPCR, and we can do molecular biology, and now with the efforts of some amazing scientists, now we're able to get structures fairly easily. And we're like children playing in the sandbox with GPCRs because they're pretty tractable, and we can test our ideas fairly quickly without having to spend 5 years learning how to run the assay.
Yeah, I think you're right. So I think working in the lab and measuring GPCR function or ligand binding or all of these things, to me they're a form of art. It's like cooking. Sometimes it works, sometimes it doesn't. And if you would talk to an electrophysiologist who'd say, of course I can do this, I can measure an ion channel activity, it's because we have never practiced it. Yeah. And sometimes with new GPCRs, it's kind of an odd way of looking at it, but I think it's just you have to develop that relationship with your receptor where you get to know them and you're like, oh, so you actually prefer these conditions for me to get the responses I need. And and yes, I think we've made tremendous progress when it comes to data acquisition, whether it's the type of assay or the speed of it or how many compounds we can test at a time. But to get back to data analysis, you can collect as much data as you want, but if you don't have the right way to analyze it and interpret it, then you're pretty much doing it for nothing.
Yeah, that's a good point, Yamina. And I think the other thing I like about GPCRs is that thanks to people like Terry Kanakin and a lot of other people, the data analysis is not that complicated. I work with other classes of targets, and enzymology. Trying to do data analysis for enzymology, you need like a 5-year PhD to learn how to do that. It's really tough. But for GPCRs, the underlying theory and the math is designed to be tractable by people who work at the bench. So, like with biased agonism, we're using the operational model to analyze the data. And you could imagine that would be something that would require specialist knowledge really to do. And it's not trivial, don't get me wrong.
But it is not. I don't. It may seem very trivial to you, but it's not. It's not to everyone. You have to do it. I think it's just a matter of practice and having the right resource like yourself, or going through Terry Kanakin's papers. And even when you go through the papers, you see all those formulas and you're like, okay, so what do I substitute and where do I plug my data to get the response?
It can be tricky. But I got to tell you, if you compare that with doing numerical solution of differential equations to solve enzyme mechanisms with the pings and the pongs, and it's like that's hell. This is easy by comparison, but the proof is in the pudding. With the biased agonism, we have hundreds of papers now being published with. Arthur Christopoulos has done some great work in here. Fred Elhert, where it's really bringing analysis to the masses. Yes. And you make a good point, Yamina. It is tough. It's not something you can just pick up in a day, but it's something you can pick up in a couple of weeks, which I think is good. I went to see one of Terry's classes once where he was teaching people how to do this and Terry's like, what are you doing here? You know this stuff. And I'm like, Terry, I'm here to learn how to teach. And I watched him in action, and, you know, there's a lot of papers now using very difficult pharmacological analysis that people have gotten used to because he's taken the time to train people how to do it.
Yeah.
I love that about GPCRs as well, is it's a very generous field, you know. And,
Yes.
and even look at your podcast, you know, so many leading experts and also just postdocs who are busy taking their time to talk to you about GPCRs. You know, we're a generous bunch in this field, and I like that.
I agree with you, and I'm very grateful for everyone who has either already come on the podcast or accepted to come on the podcast. I feel like I'm in the right field. People are generous, they're giving, they take the time because we have a common interest, which is GPCRs. And at the end of the day, working together seems the best path forward to drug these receptors.
Absolutely. Yeah, absolutely. Yeah. And
please go ahead.
Yeah, and I think now might be a good time to talk about the summit. You know, I think you know, talking of generosity, you know, you've been so generous with your time this year, just making these resources available, the podcasts, the ecosystem on the website, and then the summit was just was magnificent. You know, it was a whole new way of doing things, and I'm imagining that was a lot of work, but we're all grateful to you for that.
Thank you so much. It was a bit more work than I anticipated, but I think it was absolutely worth it because a lot of us could not travel. As you know, I don't work in the lab anymore, but it's still, if I look at it from my perspective, I was still able to watch at my own convenience these talks. And I'm grateful to all those speakers who came and gave, you know, either a live talk or pre-recorded talk. And a lot of these speakers still have their talks available on YouTube.
Yeah.
Which is a rare find. And we're already super excited working on the 2021 Dr. GPCR Summit.
Cool. Looking forward to it.
That will be phenomenal. I think I'm hoping we'll get even more talks. What one criticism or one suggestion that came in through the audience is that there was no way of interacting live. So we're going to be working on that and trying to set up, you know, every day a couple of hours maybe where people can join in live.
Yeah.
Or alternatively, it's gonna be a spoiler, but we're working on getting a once a month Dr. GPCR webinar, a live one. And I have to credit Dr. Arun Shukla for the idea.
That's a great idea.
But I think we can do it live. We can have either record. We're gonna ask the speakers if they let us record it and publish it. If not, people can sign up, come in, and then we can interact, listen to a great talk, and also interact and ask questions. I think it's an addition to the ecosystem other than the podcast, the newsletter, and the summit.
Oh, that's going to be great fun, Yamina. That's great news.
I love the idea. So we're working on it, and we're working on getting speakers to come and join us.
Cool.
Yeah, super. So you're in the UK, you're working on allosteric modulators of GPCRs, you're doing your PhD. You like to work at the bench, but you definitely prefer analyzing the data that you generate or your colleagues generate. And then what's next? How do you decide about your next career step?
You know, it was interesting. It was like the decision was kind of made for me. And I think this is, you know, you talk a lot about careers, and I don't know if it's a good thing or a bad thing, but it's a thing. In science, we have to be willing to travel. Yes. I know that can be very difficult for some people. For me, it was something I wanted to do, but I was really interested in GPCRs and structure function work there was a great job opportunity at the National Institutes of Health in Bethesda. And I was hemming and hawing about moving, but again, it was at a time when there weren't a whole lot of jobs around. You know, right now we're living in a boom time. Back then,
Yeah.
things were a lot tighter, and it was like, well, okay, the choice has been made for me, so I decided to go to NIH, and that was an unbelievably good time. You've been to NIH,
Yes.
you mean you know what it's like, but it was, it's. I was talking to Shivani, who's going there soon, and
Yeah.
I'm just so envious that she's going to get to experience it.
Yeah.
But it's yeah. I think what I learned at NIH was mentorship. That was the number one thing I learned at NIH. It's all very well knowing how to do something, but if you can't transmit that knowledge to somebody else, it's
Yeah.
that's not good. So, and I think at when I was at NIH, there was a few really formative experiences. But the first couple of projects I worked on, my supervisor during my postdoc was a scientist called Ted Usdin, and he worked in Mike Brownstein's lab.
Mm-hmm.
And Ted is an amazing bench scientist. I learned a lot of techniques from him. But after the first couple of years, I was going through a bit of a rough patch. Things weren't working, ideas weren't working out. We all go through this as scientists, and
Yep.
And I went out one day. Ted and I went out, and we went out on the river rafting. And on the way back, we just talked, and we talked about the reality of where I was at, and he basically gave me a cookie cutter molecular biology project, which was cloning a species homologue of a receptor. And
Mm-hmm.
he worked with me at the bench directly for like 2 months, and
Wow.
and it completely restored my confidence. And I kind of owe a lot of my career to Ted, because without that, I think I might have thrown it all in. After that, things really started working, and it was thrilling. The other thing about NIH is I learned that even the most senior scientists love talking with students and early career researchers. And I think it amazes me to this day. I could wander into the lab of a Nobel Prize winner and talk to them about science. These are the people that wrote the textbooks that we study in undergrad, and we had this fireside chat series where, really senior scientists would sit down for an hour with like 20 of us and just talk about science. And these would be people like Harold Varmus or Sol Snyder or Julie Axelrod, and it was transformative listening to scientists like that. And I'm amazed to this day that scientists just are perfectly happy, or that some scientists prefer talking to students than to their fellow bigwigs, and I love that. I think it's great.
One definitely trend about the NIH and my experience, and it pretty much reflects what you were saying, is that if you need help, it is so easy to find it, and people are so giving. And whether it's just a chat about something, or whether it's materials or anything you need, there is always someone in some lab somewhere on campus, which is huge in Bethesda, that will be able to help.
Yeah. Isn't that terrific?
I think it's terrific. And I had the same experience at Rockefeller University, where it's a much smaller campus. It's a smaller university as well. Mm-hmm. But, I've responded to emails to requests from random postdocs saying, well, I'm trying to run this assay. Has anyone done it before? Can you help? Mm-hmm. And it ended up being a collaboration and a publication in PNAS.
Oh, that's exciting.
So, and sometimes it just ended up being, thank you, it's working now.
Yeah.
And the answer was just add some BSA. but still, it's just that ability to communicate and to help each other, I think, just wonderful.
Yeah, absolutely.
It's unique. It's definitely unique. So what did you work on? What receptors did you work on at NIH?
Yeah, so at NIH, I started to get into Class B GPCRs. So these are, these are my favorite receptors.
That was one of my questions. There's only 15 of them.
Only 15 of them.
But they do very important things as well. So I think we can all appreciate them. Why do you favor Class B GPCRs, or why do you like them more?
I like they're fairly tractable. So they all activate GS, which is a nice, easy assay to measure.
Yeah.
Not like trying to measure G12, 13 and stuff like that. But from the big picture, they are involved in some very, very interesting biology. They're activated by peptide hormones of like 30 to 40 amino acids. And those hormones control everything from the stress response, bone formation and remodeling, glucose homeostasis through GLP-1. There's just a whole ton of biology out there that they're involved in. And the other reason I like them is because they're tough therapeutic targets. So we've developed peptides for a lot of these receptors, and they are very, very useful and beneficial drugs to a lot of people. But the trying to find small molecules for these targets has been a much bigger challenge, and I think that I really like that challenge. And they're just they're easy. Their binding mechanism is like interesting, but not so complicated that it's taken 50 years to work it out. And there's biased agonism in the family. There's constitutive activity that leads to with genetic mutations in humans that leads to disease. All of the stuff you see for the sort of beta-adrenergic receptor, you see for the PTH1 receptor, for example. Tom Gardella's lab and a lot of his colleagues, some of that work is just really illuminated what that receptor can do.
I know Tom. He's phenomenal. He's phenomenal. I've worked for a very brief time on PTHR1, and I think being a chemokine receptor person, Class A, I think it was a challenge. It was a great challenge. And reading all those papers about PTHR1 and what it does physiologically, I mean, I never thought that, the receptor controls so many things, including teething. So children who don't have their adult teeth come out, or they have the baby teeth but nothing underneath, that's a phenomenon controlled by PTHR1.
That's cool.
Which I find super cool. And there were other physiological events that were controlled by this receptor that I would have never thought. But I think hormone peptides are so fascinating.
Yeah. And I don't know how you handle the chemokines. There's just so many. And that was too much for me. I just
so I kind of fell into the chemokine realm a little bit accidentally. I don't know if anyone knows, and I have ever told you this, but initially in undergrad, I wanted to spend a summer internship in Michel Bouvier's lab.
Oh, cool.
And he had one spot, and before me receiving his response after interviewing, I knew I wasn't getting it because my colleague, Martin Audet, who's actually a professor at the University of Sherbrooke, was jumping of joy, telling everyone, Michel picked me. So, I kind of knew before getting the email, but then Michel, being the wonderful scientist and the wonderful person he is, he CC'd a new scientist, a new researcher at the Saint Justine Hospital Research Center, who was Nicolas Seveker, who he introduced me to, and he said, Yamin, I really liked you, but there was just one spot this summer in my lab. Let me introduce you to Nicolas. And Nicolas had worked on HIV, but his favorite receptors were chemokine receptors. And I thought it was such an intriguing question, having, one receptor with 3, 4, 6, 7 chemokines that could bind. And the complexity out of it is still fascinating to me. And then when I got into Tom Sackmeyer's lab at Rockefeller as a postdoc, the fact that the whole CXCR3 project started out with a conversation with Tom, who said, well, how come you have 3 splice variants of this receptor? And I said, well, I don't know. This is not what I wanted to work on, but let's just find out. And I think it's just really cool as a topic. But not to say that class B GPCRs are not cool. It's just I accidentally fell into the chemokine world and I stuck with it for a long time.
Good for you. Yeah, it's you look to be having a lot of fun on those projects, and it's amazing how you manage to extract a relatively straightforward package of information from those receptors, given how complicated they are. It was nice to be able to read those papers and understand them.
Thank you. It was fun. I'm glad that you felt through reading the papers that I had fun, because I really did have fun. It came with some frustration sometimes, but I think at the end, when you got the full picture, you've acquired all the data, you've analyzed all the data, and you kind of think about where your data is in the grand scheme of things.
Mm-hmm.
And then especially when you send it for review and reviewers come back and say, oh, what an interesting topic, you say, oh, thank you. I'm happy that I'm not the only one being interested by this topic.
Yeah. That's cool.
Yeah. All right. So let's get back to you here. So you're at NIH, class B GPCRs. You're really enjoying it. Then what? How? So you'd mentioned that you did a PhD and also a postdoc. Kind of, it was decided for you because there was no space in industry.
Yeah.
Then what did you do next?
Yeah. So at that point, I decided I really wanted industry, and because the kind of projects I was working on were becoming more and more therapeutically focused, and I also took a grant writing course at NIH.
Mm-hmm.
And, I realized this isn't going to work for me. I don't have this talent, and it was good actually. You've talked on here about careers and how sometimes you have to figure out what you're not good at to find your right path. And it was like,
yeah,
the academic path is just that's not where my talents lay. So I got my lab chief, Mike Brownstein. He was working with a BioTech company out in California, and who were trying to crack difficult GPCR targets. And one of those was a class B receptor, which is the CRF receptor, and CRF is corticotropin releasing factor, a big player in the stress response. And so the company, so I went to work for the company. I got hired, and it was a really good fit, and I was really grateful for that opportunity. And I was working with, my main role there was at the beginning was working on the CRF project, and that was headed up by a scientist called Dimitri Grigoriadis, who's a Greek guy. You won't be as you may see from that name. And he's out of Toronto, so he's part of the Canadian mafia. He worked for Phil Seaman on dopamine receptors, and so I knew his work from that. And Dimitri is just a inspirational scientist and a brilliant communicator. And so we got to work on the CRF receptor. And Dimitri's philosophy is, and not everybody shares this philosophy, is to crack a difficult target when we're trying to make a drug for it. You really have to understand how that receptor functions. You have to do the work to figure out how it works. And so my role there was, at the beginning, was really figuring out a lot of the structure function stuff. Mm-hmm. And that was with an amazing team of experimental investigators. And so I just want to make a quick shout out to the team here. And that was Dimitri, who was the project lead. Then there was Beth Fleck, who's an amazing pharmacologist, analytical pharmacologist like us. And then there was Sue Sullivan, who was an amazing experimentalist who got a lot of the assays working that we ended up using to figure out how the receptor functions. All too often, I think the people that get things working don't get enough credit, but Sue was instrumental in making that project go. Paul Crow was the biology lead, and then Brock Brown was another genius assay developer, and finally, the medicinal chemist lead was John Williams, who's kind of a legend in our little world. So it was working with them it was really at Neurocrin where I learned about working in teams. The teamwork in industry is an order of magnitude greater than the teamwork in the academic world I was living in at the time. I think times have changed now. We're doing big science, which requires us to be more team oriented in academia as well. But it was absolutely thrilling, and there's the whole CRF story that we've published on, and how we figured out how to crack that receptor, which was just great fun.
That's phenomenal. I think when it comes to teamwork, based on my recent experience, I think it's still different between academia and industry, but the goals are also very different as well between the two worlds. And I think the level of funding is also very different, and the timelines are different.
Yeah.
Because you're in a company, the goal at the end of the day is to survive, which is, yes, you want to get a drug accepted, or you want to be bought by a bigger biopharma or pharma, or you want to have a relationship with these big companies because you want to get your drug into the clinic. But at the end of the day, you need funding to do that.
Yeah.
And then you have to work together. You have potentially more resources. Things are much faster than in academia. But I still feel like it's two fields or two separate worlds that have every reason to be connected.
Yeah, absolutely.
Because they complement each other.
Absolutely. Yeah. Yeah. It is interesting. It's a bit of a culture shock when you first go into it, but it is kind of exhilarating, and
it is. And it is a culture shock. It's also, especially if you've spent quite some time like I did, and I think you did a postdoc too, and I did almost too. In academia, you see the speed at which things are going, and I think it's just phenomenally exciting to know that the goal is not to show that you can pipette, but the goal is to get the data, the most accurate, the most reliable, reproducible data, analyze it, and then move to the next step as quickly as possible.
Yeah, absolutely. Yeah. Yeah.
So yeah, I think that was great. So you spent almost 15 years at Neurocrin?
Yeah, and that was it was an incredible experience. I feel pretty lucky. It was, there were good times and bad times, as in any organization. I think that scientifically the best thing during that experience was on the CRF receptor, where we were trying to make small molecule antagonists, and our very first compound that we got into like phase 2 eventually crashed out for tox, but that compound worked pretty well in our animal models, and we were really pleased with it. And then we started to make backup compounds, and we were using just a regular old binding assay to drive our SAR. And we were getting compounds with what looked to be the same affinity as our first generation compound.
Yep.
And we put those in vivo, and they didn't do anything. And it was tough, and I think it's a testament to the leadership from Dimitri and John that we were able to get through that, because under those conditions, things can go very wrong very quickly, but they held it all together. And this is where Dimitri's philosophy of figuring out how this thing functions really paid off, in that Sue had managed to get some radioligand binding assays working using radiolabeled small molecule drugs rather than the peptides, and, which was really a real challenge, but she got it working. And then Beth one day was measuring the dissociation rates of the radioligands, and it just so happened that the ones we were testing, one of the radioligands was our first clinical candidate that worked really well in vivo, and one of the molecules was one that didn't work very well at all. And John Williams was kind of instrumental in forcing us to confront that reality. It's like, this compound doesn't work, guys. You need to figure out why. And we discovered that the one that works had a really long residence time on the receptor.
Ah.
So its dissociation rate, you measured it at room temperature and it didn't come off after 7 hours. There was just no dissociation
Oh,
for 7 hours. Where
that's a long time to not come off.
yeah.
I thought you were going to say 7 minutes or 70 minutes, but it's 7 hours.
Yeah, it was incredible. And the one that didn't work in vivo came off in 15 minutes. So it was like
interesting.
And so that was, you talk about an aha moment. That was a team aha moment. We were all sitting in that meeting looking at the data going, that is a potential explanation. So we had to do the work to really convince ourselves that that was the true reason why. There's a lot of variables in drug discovery, as you know, Yamina.
Yeah.
And we we convinced ourselves that it was largely due to the dissociation rate. And then we put in place assays to actually find compounds that had a slow dissociation rate. So we understood how the receptor functions, as Dimitri was suggesting. We were able to use that to solve a problem in drug discovery, one of the biggest problems we ever face, which is compounds not working in vivo.
Yeah.
And then we were able to turn that around and use it to find new molecules. We were able to do SAR by off rate. And this was back in like the early 2000s, before this stuff became more well known. And we got compounds and we advanced one to the clinic, and it worked in phase 2. So it was really a really gratifying scientific story. And we wrote this up and as a case study in Drug Discovery Today last year, if anybody wants to read more about it. But that was incredibly rewarding. And Neurocrin are still in this space today. They have yet another compound that they're taking into phase 3 at the moment. Wow. So that was really satisfying.
I love the fact that you really wanted to take the compound into the clinic, or in phase 3 trials, but then the fact that all of you took the time, and this is what I was talking about when I said you have to get to know your receptor and develop that relationship. And this is exactly what I was thinking about, where you need to understand how that particular receptor works, and really the structure function relationship is crucial. Although sometimes receptors are from the same family, they're all unique and different, because otherwise why would 2 of them exist?
Absolutely. Yeah.
And I love it. And then you had a team of chemists who could very quote unquote easily and quickly make modifications to this compound that was stuck on the receptor for 7 hours. I still cannot believe it. 7 hours. That's a lot of time.
Yeah, but Beth applied this technology to a different project, and she was able to get compounds that had dissociation rates of like 24 hours at 37 degrees.
Oh God.
That's like she took it to insane places, and it was the whole off rate story is an interesting one. There's nuances, as there is to everything, and it's been through the sort of like hype and bust cycle now, and so now we're able just to treat it as a tool, which is good.
Absolutely. I think this is the type of information that you need to understand before you go in vivo, or before you measure, signaling effects, because you really want to know what's going on at the binding. And it may be kind of a sequential way of looking at it. So the way sometimes I see these things is I want to know, does it bind? What's the affinity? What's the off rate? And then, okay, if it binds, does it activate any number of pathways? And if it does, okay, is there a preference?
Yeah.
And then try and see what happens in vivo, in mice or rats or an animal model, and then try to correct some kind of a pathophysiological event. And it may be just really a simplistic way of looking at it, but if you don't know if you don't know that the molecule binds, or the mechanism by which it binds and how long it stays on the receptor, it's very hard to have the complete picture.
Yeah, absolutely. I couldn't agree more. And there's another project I worked on, which wasn't a GPCR. It was a transporter, VMAT2. It's a vesicular transporter. So,
Okay.
The pH of intracellular vesicles is pH 5.5.
Yes.
We had the same, and this is we had a similar issue in that some of the compounds we were making were active in our in vitro binding assay, but they weren't working at in vivo. And of course, like everybody else, we were running our in vitro binding assay at pH 7.4, physiological. And, these compounds work great. They bound with nanomolar affinity at pH 7.4. And then we decided, that vesicle, somebody mentioned, that vesicle's acidic. Maybe you should try it. We did the assay at pH 5.5 and the compounds were dead. Sometimes,
Yeah.
I think the job of the pharmacologist oftentimes is to solve problems. It's especially in industry. And particularly in my sort of newer role as a consultant, we have, you and I, Yamina, we've worked at this a long enough time to know where all the pitfalls are. So we're able to solve problems.
Yes, because we went through all these problems ourselves. And then we needed some encouragement. The way I always saw it, and this was my PhD mentor, Nikolaus, who's always said, when we had a dry spell and things just fell apart and just stopped working. And there was 2 consecutive summers where in the lab we were unable to produce DNA, to purify DNA.
My goodness.
And then when you're trying to transfect something into cells and look at signaling, you need the DNA. And so we've went through all the troubleshooting, but it never worked. And miraculously in September, this was, I think, from June to September, it didn't work. We did every possible thing. And then in September, we went back to the original protocol, just the way we were doing it, and it worked. And it's a mystery in science, but he always said, think of it as you walking through the desert, and then that there is an oasis somewhere. Just go ahead and continue.
That's great advice.
I've been there too, and I think I think it's just a matter of experience, and also experiencing the failures, and also experiencing the successes.
Yeah.
But it's not it's not the destination, it's the path that you go through that gives you that experience.
Absolutely. Yeah.
So then 15 years at Neurocrin, and I know you mentioned why you decided that you were more attracted to data analysis, and this being a consultant was allowing you to help others analyze their data. Was there an aha moment or a moment in your career at Neurocrin where you said, what, I just want to analyze the data and that's it? Did something trigger it?
That's an interesting question. I've never thought of it that way. I think it was a series of drips. It's how it is, and part of it was, again, knowing that my time was coming up because I couldn't keep up with the technological developments, and and my buddy Steve Perry, who's like really technical, he started out as a GPCR pharmacologist in Bob Lefkowitz's lab. And now he's running gene therapy programs. He's that guy.
Wow.
But I knew I knew I wasn't going to be able to do that. But the other thing is, I made a point of getting out of. I had an office. I had all the usual things, and I was talking to a young guy, Derek Meow, who's working in there, and he's like, get rid of your desk, get rid of your office, get out, you got to talk to people. And he's one of these younger people that thinks this way. And so I took his advice seriously and just set up my computer on my lab bench, got rid of all the paper, and put my lab bench right in the middle of the most trafficked area of the lab, where everybody had to walk through to get to the office. And I just started talking with people, and I was doing that already, obviously, but I became the person that people went to solve problems. They'd have a piece of data and they'd say, I can't figure this out. What the heck's going on here? Or they come with a new project idea, and and I'm as wrong as often as I'm right, but it's always good to talk. So I got into the habit of sort of just helping people in the company.
Mm-hmm.
And I really like the problem solving aspect of it. A lot of people, you were talking about the DNA purification problems and everything like that. It's like a lot of people don't really enjoy troubleshooting, and that's normal. I guess I'm a little abnormal, and I really enjoy that troubleshooting, and trying to figure out why something isn't working and to get it working again as quickly as possible. So I was doing a lot of troubleshooting work as well. And then finally, there's just the data analysis. Dimitri once said, I rely on you to make sure that the data is done right. So I was just doing more and more and more data analysis, and some of that was fairly sophisticated, like the kinetics and stuff like that.
Yeah.
So that was like, and I was like, and then there's another couple of things. People from other companies would call me up saying, I've got this really weird data. Can you help me out looking at it? And I was like, I kind of can't because I work for a company, but so, and then I got interested in a research problem as well, and I was, and that research problem was signaling kinetics and how we can quantify it.
Yeah,
which is something I've been working on a lot. So all of these things coalesced, and it was like, you know what? And then something personal happened in my life where it was like I had an opportunity to move, and it was like, you know what? It's time to move on. And it was a tough decision, but it was an obvious one at the end of the day. So from there, I went to set up the consultancy company PharmaChanics, which is, that was a good move.
I love the name. I really love the name, and I think you touched on so many important points, which include knowing your limits, knowing what you like to do, knowing or discovering or exploring at least what you don't like to do and what you're actually not that good at. Some people are amazing at everything they do, which I'm pretty sure it's a small percentage. I was talking to Michel Bouvier earlier this week, and he was talking about Professor Julie Axelrod, who always went in with the newspaper under the arm to the lab at 9 and then walked out at 5 with the newspaper under the arm, and that was it. And then Michel was mentioning that he is not that smart. He works hard. And some of us are very smart, and some of us need to work hard, and some of us just are not very good at everything. They're just good at a certain number of things. And I think this is true for everyone in the world, not only in science.
Yeah.
And if I had a message to the audience, especially to postdocs and PhDs, don't beat yourself up on the things that you don't know how to do. Try it. And if it doesn't work, just focus on what you're really good at, because most likely you're in that rare number of people who actually are really good at that.
Yeah, you mean it. You're totally right. And I like that about this podcast, that, you're talking to early career people who, we read it on Twitter, a 22-year-old PhD student is terrified that they're not going to be able to get grants. It's like, chances are you're going to be doing something very different, and it's going to be a lot more exciting for you.
Exactly. And I speak of experience. I beat myself up because, I don't know, a Western blot didn't work, but then I could magically make work a cyclic AMP in plate transfected assay, and the cells were just beautiful, and the dose response curve was magnificent.
Yeah.
But I wasn't focusing on the fact that I was able to do this and acknowledging that it's unique. Not everyone knows how to do this. I'm not saying I was the only one, but there was a large percentage of people who may or may not be able to do this experiment. Yeah. And then, I should have focused on the fact that this works, and let's try and get better on the Western blot. Yeah. Or find someone who's really good at the Western blot and just collaborate.
Exactly. Yeah.
So, but did you find, going into consulting like a big leap of faith? Because I listened to a lot of podcasts, including one about consulting, and a lot of times consultants in general, not necessarily in science, they have these periods of abundance and they have these periods where there is not enough work. So, and it's scary to people.
Yeah, it is a bit feast and famine. But to me, I think I've found something that really suits me, so I'm able to get through it. Consulting is helping people solve problems, and I enjoy helping people, and I enjoy solving problems. So it really kind of suits my personality. So I can, through the difficult times, if you know what you're doing is what you're supposed to be doing, it makes it a heck of a lot easier. It takes away the self-doubt, and you kind of know that stuff's going to work out. It takes a certain amount of faith, and I had to learn how to do a ton of stuff. I'm not a good verbal communicator. I'm a math person, right? So I can analyze the data, but some of my early papers are it's like they were written by the Unabomber. They're so complicated that nobody can understand them. But when I was with Neurocrin, when I was working with Dimitri, we would have this. We published a bunch of papers together. It was phenomenal, and I would do the math technical side, and Dimitri, who is a genius communicator, kind of like you are, Yamina, just born communicator, would take that information and then put it in the context of what the audience wants to read, what the audience wants to hear. When I started up on my own, I didn't have Dimitri to do that for me anymore, so I had to learn how to do it myself. And so it's never going to be my it's not something I was born to do, but it's something I have to do to do the job. And it's the generosity of people. I've been able to talk to people who've been able to help me understand how to do that. Dimitri's been very helpful. You've been helpful, Yamina. A lot of our conversations have really helped. And then anybody who's sort of set up their own business is very, very enthusiastic to help you set up yours. People are so generous with their time. So, yeah, it's I had a sort of natural aptitude for consulting, but I had to really get much, much better at communication. That's been absolutely essential.
Which, when you think about it, it's a learnable skill, and I think it's a little bit like confidence as well. I think it takes a lot of confidence, and you may not feel it, but I think you have a lot of confidence into saying, you know what, this is what I like to do. These are the things I know how to do. These are the things I need to learn. But at the end of the day, I love doing this, so I'm gonna go, keep the faith and just do it. And I think confidence as well is also a learned skill. You're not born with confidence.
No.
You acquire it over time. I mean, I have a 5-year-old, as you know, and I will never forget the first 2 months where, as new parents, we just didn't know what to do. But you could tell that the child was like, what am I doing here? Why is there so much light? And why do I have to actually work to eat now? And this is the image that I have, is that you just build that confidence over time, and you just have to go out there and put yourself out there. And you're right. I think people in general are wanting to help because your success will lead to their success as well.
Yeah.
And I think you get much more out of collaborating and out of, making referrals or, having finder fees or just having a conversation that's gonna, and again, it may sound too optimistic or naive if you want to put it that way, but I think that's the best way of looking at it.
Yeah, absolutely.
Going for it, and what does it cost you? 5 minutes, a kind word, or just, sharing an idea at one point is just amazing.
Yeah, absolutely. And people are so generous. And the other nice thing about sort of being your own boss, if you like, is I don't know how you'd feel about this actually, but I find that it's a lot easier to make mistakes, which is a good thing, which is a really good thing. I'm able to try things. Some things don't work. Now, trying to do that when you're reporting to somebody and you've got something to lose can be difficult unless you've got a really good boss. But I love just setting up your own, doing your own thing. Is you have the freedom to fail, which is awesome.
Yeah. Yes, absolutely. I think it makes a lot of sense, and especially if you don't have a good mentor, you don't have a good manager, and unfortunately if you have a manager, it can be very difficult because sometimes communications fail with your manager, and then you think you're doing the right thing, or even if you ask for feedback, you don't get the right feedback. But being your own boss, it's, you know what, it's, if you're successful, it's thanks to you. If you're not successful, it's your fault.
Yes.
And you better find a way to finding the path that would lead you to success. It's kind of losing that that safety net.
Yeah.
Or making your own safety net somehow.
Yeah, and I think the safety net are all the other people who are doing the same thing as you are. People like, it's great to talk to you sometimes, you meet up about some of the things that haven't worked. We can commiserate with one another, but then on the other side, there's all this stuff that you find out you're good at stuff that you never knew you were good at.
Yes.
That is, like making videos. I'm not sure I'm not Steven Spielberg, but I've discovered that I can communicate really well with training videos and stuff like that. Never would have discovered that if I hadn't taken the leap of faith to try.
Yeah, and I think it's it can be scary trying new things. It's scary when you don't have that safety net. But because I think the way over time and culturally, the way that things go is that you're born, you go to school, you get a degree, and then you go to work for someone.
Yeah.
And 20, 30, 40 years ago, you worked at the same place from 19 to 65.
Yeah.
And now you jump hoops, you jump jobs, but that model is not for everyone.
No.
And it's hard to get out of that model because that's what you've seen. That's what most likely your parents went through, your grandparents went through.
Yeah.
But, sometimes telling yourself, getting let go from a job is it's at the same time the worst and the best thing that can happen to you.
Yeah. Yeah.
Because everything that life throws at you on the long term, I'm getting really philosophical here and very far from GPCRs, but you can do it. If in my mind, if something gets thrown at me and I find it difficult, it's because I know that at some point I need to solve a problem.
Yeah.
I need to learn something new to get through that.
Yeah, and that's all about confidence, isn't it? Like you're getting, that's how you learn and that's how you get confident.
Exactly. And don't get me wrong, it's the hardest thing ever.
Yeah.
When you're in that, deep end of the desert, like I referred to before, and you're like, oh my God, which way should I be going? I can't go back.
Yeah.
And the only way is forward, but then you're going to get thirsty, you're going to get sunburned, and you're hot. But at the end of the day, you have no choice.
Yeah.
Just to move forward.
Yeah, that's true.
So what other things did you learn since you started being a consultant? So you said you learned to communicate better, mm-hmm. and you've had a lot of help. What other new things did you have to learn?
I had to learn how to collaborate, funnily enough. And that's been, and it's like the different organizations have very different cultures. Mm-hmm. And some of those cultures I blend really well with. I'd like to think I blend with most of them fairly well, but some working cultures I kind of don't. But mm-hmm. Like a really good example is I have just this wonderful working relationship with Montana Molecular, which is headed up by Anne-Marie Quinn, and I get to work with Thomas Hughes and Paul Tucson, who are just amazing scientists. And but I had to learn how to collaborate. It's like I think for most academics this comes fairly naturally, but I had to learn the give and take. And the person who sort of taught me how to collaborate really was Anne-Marie. She's an amazing collaborator. And,
she's wonderful. She's just wonderful.
the patience, the open-mindedness, the emotional intelligence. I guess that's one thing. I guess that's something I didn't realize I had. But to be a consultant, you have to have a certain amount of emotional intelligence, because when people are coming to you with a problem, that's not a nice place to be in, right? You talked about your DNA stuff, and it's like it can be, we get better at handling problems over time, but
Yeah.
And sometimes, as being a consultant, the most important thing is to be positive, and to be fun. I was helping out a company this year. I won't go into details, but they were, as we often do as scientists, they were getting really wrapped around the axle about this data. It's like there's something wrong with this data. This doesn't match that. That doesn't match this. And they were talking to me. They got me on the phone. It was like, you know about this. Can you give us some help? And we started talking, and and they were just a great crowd, and we were able to relax. And as we were talking through the data, all I was in this process really was just a sounding board. They suddenly realized that these huge problems that they thought they had weren't actually that big problems. And they were pretty manageable, and that the differences they were seeing were actually in some cases smaller than the normal average. So it's that, that emotional intelligence is that's something I had in me, and by working with people like Anne Marie and Terry, I was able to see that I had that too, and that was a really pleasant revelation.
And you had to go and become a consultant to learn that about yourself. So again, it goes back to that confidence and the fact that sometimes you go on this discovery journey and you discover things about yourself even after you're 20 years old.
Yes, exactly.
which I have been 25 plus for quite some time now. But yeah, I think it's that. And I love the fact that, as a consultant, I've not done that, but the way you were mentioning it is that you are that sounding board. You are that expert who knows a certain amount of things, but then when people get stuck in their problem, it's like this wheel of overthinking things. You're there to kind of untangle that web and really guide companies and guide customers, clients through that process and tell them, okay, this is the problem that you're seeing. Actually, let's try and find another way of looking at it.
Absolutely. Yeah. And that's that can be occasionally that can be quite challenging, because it's very easy as a consultant to forget that the client is dealing with a very complicated set of interlocking parts when they're working on a project.
Yeah.
When you bring a consultant in, I'm only seeing a very small piece of that puzzle, and I have to remember that what might be a very clear and obvious solution to me is actually not feasible because of a technical constraint I'm not aware of, or because of timing constraint. All of the rest of the stuff that person on the call is having to deal with. So I have to be, I made a few mistakes early on where I just was not sensitive to that reality. That the client has got a lot of pressures that I'm not aware of. So over time, that's something you learn, right? It's like different people have very different outlooks and priorities and perspectives, and so part of the challenge of being a consultant is just being sensitive enough to that reality,
Mm-hmm. Yeah, and I think you made a great point about talking to clients, is that sometimes you need to figure out what is the right question to ask, because if you want to provide the right solution, or at least give the advice that will lead them to solve the problem, you have to have a complete picture.
Yeah.
And sometimes they miss a detail, because to them it's just a detail, or they know this, but then you need that information to provide the correct advice.
Yeah. But then, on the upside is when, maybe half the time you solve the problem really fast. They're calling you because you have an expertise
Yeah.
and, that they don't have. And they lay the data out in front of you and you're like, oh, it's that. And they're like, oh, we didn't know that. We didn't see that before. And it becomes immediately obvious. So that is, you get a few of those, and that's really fun. When that happens, that's very gratifying.
Yeah, it takes you back to the time when things haven't been working for 2 months, and you're like, yeah, this is what I learned from that. So usually your clients are in the GPCR field, or is there more of data analysis and enzymology oriented projects as well that you take on?
So my clients, that's an interesting question. They are a lot of them are GPCRs. So particularly my Class B GPCR expertise has gotten a lot of it's kinetics. So as you know, sort of my research obsession for the last decade has been kinetics. So a lot of people come to me for help with that, and that's really rewarding, working on kinetics with people. And so I've done with Montana Molecular, we've done some pretty extensive studies on quantifying signaling kinetics for GPCRs. That's been hugely rewarding. And then there's also just some companies want me to help them interpret the data they've generated. So it's like, this data isn't making sense. Can you help us make sense of it? And sometimes that's not for GPCRs at all.
Okay.
I've dabbled in enzymology a bit with a company called Assay Quant. We're working together to sort of do some streamlined data analysis to make it more rigorous on the one hand and faster and more reproducible on the other. And that's been a heavy lift, but it's been incredibly rewarding. We've done some pretty interesting things together. And then, but beyond the consulting, then I also sort of like do my own side gigs as well, my own scientific research, and that's a whole other sort of that's beyond. That's not really a consulting question. That's more like my sort of current research activities question, if you know what I mean.
Can you tell us more about that? I didn't know that you had a side gigs.
Yeah. So yeah, I've been. I'm really interested in signaling kinetics. We published a few papers over the last few years on this. For GPCRs, we have developed these amazing tools to measure things over time, right?
Yes.
You know this, like the Brett Biosciences for measuring arrest and recruitment and stuff like this.
Yep. I love the idea of measuring things that happen over time, and we've talked about this previously, and I think it's just phenomenal.
Yeah, and it's I think it's for me, I read like Michelle's papers, and there's Martin Loos as well. Some of the some real Mark von Zastro, some real pioneers in working out, Tom Gardella as well, on working out this spatiotemporal signaling that GPCRs can signal once we're in the endosome and all that. And I was looking at these papers, and every time there's a time course graph, and sometimes it goes up and stays up, sometimes it goes up and comes down, sometimes it goes up, comes down and plateaus, and there is and at the time there's never a curve fit to these data, and it was like, that's interesting. I wonder if we can figure that out. Imagine doing pharmacology dose response experiments for 20 years without applying the sigmoid curve fit to the equation to determine the EC50. To me, maybe I'm a bit of an obsessive, but to me that seemed like a problem we needed to fix. So so working with I had the luxury of collaborating. I had to learn how to do that with some amazing labs to really try and work out an analysis framework to analyze kinetic signaling data. And what we've come up with is something that I think is fairly easy for people to use. Not totally simple, but if you can do a dose response analysis, you can probably do a time course analysis using these equations we've developed. And the really, I think what's been incredibly rewarding about that is I made a decision early on to target early career researchers with this stuff. You know, it wasn't enough to write a really complicated technical paper that was going to go in Journal of Theoretical Biology that nobody would read. That's my old way of doing things. And
yeah.
through working with people who are great communicators, I figured out a way to publish this stuff so that people could actually use it in a practical sense. You know, people like you, people like me when I was a postdoc, who could just go to Prism, load the equation, which is something we've worked hard to make as easy as possible, and then just analyze the data. And now, you know, it's incredibly rewarding to see some early career researchers not only use it but question it and take it in new directions, and that's been incredibly rewarding.
It's a great side gig. So would this be a favorite aspect of GPCR research in your case?
Yeah, for me it is. To be very personal about it, you know, I just find time very, very interesting, sort of almost philosophically. But I like, unlike most people, I find the fact that when you add the time dimension, things get very counterintuitive. I kind of like that. And it's, but it's, I have to say, you know, GPCR pharmacology has developed drugs for a century without using time as a variable. So it's not like we have to do it. But, you know, I think, you know, it's particularly with the new discoveries of persistent signaling and how that could translate to more efficacious drugs with fewer side effects, you know, it's, you know, adding time to the equation could help us solve problems, which
faster. Yeah, I think it's a matter, and it's funny because you were talking about you know, time philosophically, and I, you know, the concept of being in the flow state. And data analysis actually always got me in that flow state when hours passed by, and I was just trying to combine and see, okay, if we look at this versus that, and, you know, having 5 or 6 or 7 chemokines, and then looking at one specific time versus the other. And I think that's, and it's not only rewarding as an experience, but also the fact that we're looking at analyzing data as a function of time can also speed up drug discovery and better understanding how these compounds or these receptors work.
Yeah. I mean, just looking at your data for the chemokine receptors, where, you know, at the activity, the relative activity of this bewildering diversity of chemokines you work with, the relative activity is dependent on the time point you measure it. You know,
yes,
and that's both exciting and slightly scary. But,
yes,
but you know, again, yeah, and Anne-Marie makes the point of, it's just something as simple as this. You know, you measure a calcium transient for a GPCR. You know, if you measure that at 30 seconds, great, your receptor couples to calcium. If you wait 10 minutes to measure that response, it's gone. So you're not even going to be able to see it. So
yes. Terry Kinnakin made a great point. So I was at his course last year at the Discovery on Target here in Boston. And the whole course was about studying GPCRs and better understanding GPCR function in drug discovery. And he was mentioning that, you know, when you're screening thousands and hundreds of thousands of ligands, you want to screen really quickly, and for a great period of time, the tool of choice was calcium flux.
Yeah.
And that gave, you know, so many false positives and false negatives because it may have not been, or it was not the right pathway to screen.
Yeah, absolutely.
Because of that signal that's very quick and transient. And it's by the time you realize that it happened, it's already gone.
Yeah, absolutely. Yeah. And I know he's a big advocate. I remember his paper showing, if you use IP1 instead of calcium, you can
Exactly.
record things much later, and your data gets a lot more robust. It's
Exactly. Exactly. But I think we've moved past that technological, issues where you had only one way of doing it, which was calcium.
Yeah.
I think we're way past that with robots and with all of these and all these various assays, including the biosensors that are now available. We can do a fairly, rapidly acquire data for different pathways, different compounds. And then we have you to analyze and help us analyze this over time and see what happens as a function of time. Yeah. So I know we've touched a little bit about this, but and I think the whole podcast, the whole discussion was a lot about GPCRs, but also a lot about advice to junior scientists. Is there anything else you think we could tell the audience who are, PhD students and postdocs who are thinking about, staying in the field, contributing to the field, but not necessarily in a more traditional way, which is, going for an academic position, but more going for an industry position or maybe into consulting?
Yeah, I think this is a really interesting topic, Yamina, and I know it's close to your heart. I think one thing I'd recommend is to be aware that we're living in a boom time for biomedical research. We are living in, you and I are old enough.
25 plus, as I mentioned.
To kind of realize, when I was first started out doing drug development 20 years ago, we were just making another beta agonist, or we were making another SSRI, and it was like, what are we doing? And now we're doing gene therapy and we're making a vaccine in 10 months.
Yeah.
This is an incredible time to be in biomedical science. Whether it's GPCRs, which this is a very exciting time. And however anxious we are about our careers, and I don't want to minimize that anxiety, you and I have felt it. This is fun. This is fun. What we're getting to do now for GPCRs in particular. Now we have the structures, we have the biosensors to tease things apart. I was reading some papers by Bradley Hoare, who's a ECR, and he was using all these incredible new techniques, and you could just see, man, this is so much fun. So, we're riding a wave here, so enjoy it. And then the other thing I think, really, I'm just going to check my notes actually.
Of course, but I'm looking at the notes as well. So the audience has to know that we use what I sometimes fail to disclose that although these conversations are never scripted, but we always have some topics that we want to talk about so that the podcast comes out as constructive as possible to the audience, as well as such a great conversation between me and the guests. And I think so far I've very much enjoyed it. I'm looking at the clock and I'm like, let's not stop, let's just go at it. I think I've received a couple of comments when people were saying, 1 hour 1 hour podcast is pretty long, but you know what, if you have time and there is important topics you need to go and talk about, then you just can pause the podcast. You can listen to it later, but we're going to continue talking to really go around the entire topic.
That's awesome, Yamina. You do an amazing job with this. I'm grateful for all the prep you gave me. It's fantastic.
Thank you.
Yes, I think I've got 2 more points. The first one is a short one, which is don't be afraid to talk to leading scientists. We see on Twitter people saying, how do I write an email to this person? And it can be daunting, but be aware that most leading scientists will be delighted to talk to you. And I'll share a very quick story, which is I was at a conference a couple of years ago, and a leading scientist was there, on GPCRs, and he gave a presentation. And at the end of the meeting, this must have been an undergrad, maybe 19, 20 years old, comes along and completely naive about the field, and asks, completely naive about who this person is, and starts asking them a naive question. And this leading scientist had other leading scientists to talk to, but instead he answered this person's question. And I thought, oh, that's really cool. That's really cool. Good. That's the way the world should work. And half an hour later, I came back, and half an hour later, he was still talking to this student.
Wow.
And it was clear that he was absolutely delighted to be doing that. And pretty much every scientist I know is the same way. There are some exceptions, and obviously you can catch somebody when they're busy, but never hesitate to talk to a leading scientist if you can pluck up the courage. They get a lot out of it talking to you as much as you get out of talking to them. And then I think the other point I had was realize, and this is really something you talk about a lot, Yamina, you're the expert on this more than I am, I think, which is realize that there's an ocean of career options out there. Sometimes to me it seems like it's easier to become an astronaut than it is to become a tenured professor.
I agree with you on that one.
And that the reality is very few people have the combination of specific talents and personality traits necessary to become an academic. And it's highly likely that your talents and your personality is going to be better suited to something else. You've had career coaches on here, Yamina, and this is something that's dear to your heart. But once you've got a PhD, there are so many things you can do within science, and it's only getting better. It's like before you used to have a choice if you're a GPCR person of working in being an academic or working for a drug company. Now you have the option of working, obviously those are still options, but you have biotechs, you have technology companies, you have assay development companies, you have companies like Montana Molecular and Domain who are developing assays for people to use, you have scientific communication. There's just so many things you can do, and you're probably, when you're starting your PhD, when you're starting your postdoc, you probably don't know what those are yet.
Exactly. You could even, become a patent agent and then go to law school and work at the junction of, filing patents that need scientific expertise. If you cannot understand the science behind the legal document, then, what use are you for? So I think having that PhD gives you the ability to think, gives you the ability to understand science, not necessarily only GPCRs. I've recently been in a situation where I needed to understand how insulin was produced. And I went in and dug into all of these. So now I know how insulin is produced. And I was trying to figure out how long. So you store insulin, obviously, in these vesicles, and then you secrete them whenever your glucose levels are high in the blood. But then what I was trying to figure out, and I actually couldn't find it yet, is once you've depleted the insulin from your storage vesicles, how long does it take for your body to produce new insulin that gets excreted? I don't know the answer to that, but my guess is it's not a long process, because you can allow the body to have to be flooded with large amounts of glucose in the blood. But I've never worked on hormones or insulin or anything, but having that expertise allowed me to go in and read that paper, and being able to understand the mechanisms and also understand the pitfalls of what do we know and what don't we know.
So that PhD has taught you how to think, taught you how to research.
Exactly. And you don't have to continue working forever at the bench. You can use your talents elsewhere. I think it's just a matter of finding the talents that you're really good at and what you really like to do, And as you mentioned, I think the biotech and biopharma scene is booming.
Yeah. Yeah.
And it's just, I mean, as you know, I live in the Boston area, and it's just grab and go. There is in Cambridge, Massachusetts, there isn't a square foot where you can find 10 companies, and one year one person is working at this company, and the next year they're working across the street. But there is there is the funding, there is the speed, there is the technology that out there. So you can work in different fields, immuno-oncology, you can work in gene therapy, you can work at companies like Pfizer or BioTech in Europe, or Moderna, or other companies that use novel technologies like mRNA vaccines.
Yeah, it's incredible, isn't it?
I find it totally incredible. I've been following, how these things work, and it's just phenomenal.
Yeah. And then there's the other, some other stuff, it's like working for the FDA,
Yes.
that you're seeing right now just how incredibly important, or working for the CDC, it's,
Yes.
Those are incredibly rewarding jobs that are very demanding and do amazing work. And
also very different from academia and industry. Yeah. And I'll have a quick follow-up question on that. But I think they're rewarding. They're very different. And if you want to learn about anything in any field or any type of job, as you mentioned, Sam, just find the right people. Go on LinkedIn, go online, just Google it, and email these people and ask them, say who you are, say why you want to talk to them. And if it's not person number one, then move on, because sometimes it's not that they don't want to talk to you, they just don't have the time.
Yeah.
I've been trying to schedule, and Michelle's going to hate me for this, but I've been trying to schedule the podcast recording with Michelle Bouvier, which I did this week I think in July. But Michelle is so busy that we finally found that time now. And I know that it's not personal. I just know that he has so many things to do.
Absolutely.
And you just, sometimes it's just a matter of timing. Just go out there and talk to people. Yeah. You won't know until you talk to people. You cannot expect to like or dislike a job if you haven't the faintest idea of what it encounters.
Absolutely. Yeah.
And then gets me to one of my questions, is actually one of your questions that you suggested, is how does GPCR science and industry differ from academia? I think we touched a little bit on it, but if we can go more deep into it.
Yeah. So we talked about the sort of cultural differences. I mean, if you like being a team player, then, it's good. The specifically for GPCRs, I guess you're asking, is the biggest shock for me when I got to industry was realizing that in vitro pharmacology didn't rule the world. The people who make drugs are medicinal chemists, and they have two questions. Does the drug work in vivo, and what's the safety margin? And everything we do, from the smallest test tube assay to doing a phase 3 clinical trial, is dedicated to answering those two questions. Does it work, and how safe is it? And so for me, my training in GPCR pharmacology in industry was about solving problems. It was always about solving problems. And that happened in two ways. The first way was, and I was lucky to be working with Dimitri, who really believed in this idea, was to spend some time researching the target itself. And we were, I was even given a number. You're expected to spend 20% of your time researching your target. And then the other side of that was simply solving the problems that come up. And the biggest one we had was, why is this drug not working in vivo? Mm-hmm. And that's where the GPCR pharmacologist really kind of earns their pay, is in solving the big questions about why compounds that chemists are making are not working, or why they work really well in this assay but not in that assay. Yeah. And that's where, if you're a problem solver like me, that's really attractive. There are other differences. Obviously, in academia, within the constraints of the funding system, you can kind of set your own project objectives.
Yeah.
You can't do that in industry. And if you're somebody that wants to work on the same target for the next 20 years, that's not going to happen in industry. What happens in industry is something wonderfully different, which is if you're willing to adapt, you learn an incredible amount. So I'm a GPCR pharmacologist that learned about neuroscience, that learned about stress, that learned about obscure movement disorders that are not that obscure, that affect large numbers of people. I didn't even know what they were until I went to industry. I learned about pharmacokinetics, drug safety. I learned a ton of chemistry from working with chemists. I've never made a compound, but just understanding how they interact with receptors, I learned from chemists. And in industry, I was forced to adapt. So I learned an incredible amount incredibly quickly. And you mentioned some of the people you're going to be having on the podcast, and
Yep.
It's like one, they spend 2 years working on immunology, and then all of a sudden they transition into working on cancer or CNS or inflammation. If you want to learn and be exposed to tons and tons of new things, industry works well. If that isn't your thing, and it certainly isn't a thing for a lot of people, industry will be a challenge.
Yep.
The other difference is, and I think too big of a deal is made of this, I really do, is there is a difference in funding level.
Yep.
And if you're at the extremes, but I think academic science these days is funded much better than it was, and it's a lot cheaper to do. Things have become a bit easier, but in industry you will have a bigger budget. I think it's public domain this, so I can talk about it. But I was involved in doing some animal studies on testing a drug for antipsychotic efficacy.
Mm-hmm.
And it was this VMAT2 inhibitor. And we published posters on this. And, and we spent $500,000 on outsourcing in vivo studies to psychogenics in a little over 18 months. So that's that's a real plus of industries. If you can persuade people to get behind your idea, you can get things done really quickly.
Yeah, I like that, and I would love to echo that. I mean, having spent some time in biotech, not too long, but just enough to realize that how different it is from the academic system. Spending $30,000 on compounds, and then $30,000 more on an instrument that at the end of the day will save you 2 or 3 months worth of work, and it's hands off compared to you being there pipetting. That is a drop in the bucket in industry.
Yeah.
Because the priority there is to get data as quickly as possible, but as reliable as possible at the same time. So the $200 antibody is nothing compared. Plus, I think the salary as well as that you take home as a scientist in a biotech company, you're an expensive asset.
Yeah. Yeah.
And so they will do everything in their power to give you all the tools that you need so that they get their return on investment. And you're an investment. The compounds are an investment. The in vivo study is an investment. And at the end of the day, what's important is the result.
Absolutely. Yeah. Time is more important than money sometimes, isn't it?
Yes, it is. I mean, look at the COVID vaccine. I cannot imagine the speed at which, Pfizer/BioNTech and Moderna have been going at, and it's just exhilarating. But when they get there and they're very close, then I'd say, you know what, they've done an amazing job, but without putting in that effort.
Yeah.
And that large amount of money. And look, especially with COVID, the fascinating thing is that the logistics behind distributing the vaccine is very complex.
Yeah.
But I can only imagine how much work and how much time and how much money went into developing these vaccine candidates. testing them, and saying, well, you know what, we're not going to wait for the approval to start producing millions of doses.
Yeah.
And we're going to already start, and it's something trivial as you'd think dry ice
Yeah.
is a problem. But then it's worth it at the end of the day, not only to, combat COVID and get vaccines out, and then we can hopefully get back to normal, but also financially, when you look at it, the first company that gets there,
Yeah.
that you can never take away from them because they're the first.
Absolutely. Yeah. It's impressive. It really is.
And so on a smaller dimension, when it comes to the GPCR field, I think that's at least that's the way I see it when you look at it from a perspective of an industry or of biotech company or of biopharma.
Yeah, absolutely. Yeah. Yeah.
Super. So we've talked a little bit about the differences in industry, academia. So one thing that I've learned from Brian Arian and a little bit from you too is that, and you just mentioned it, that when you work in industry, you can't spend 20 years on the same project, on the same receptor. How have the discoveries of GPCR function, namely biased agonism, kinetics, and things like that, affected drug discovery programs in industry?
Yeah, I think, I'd sort of take a step back for a moment. And I think for a lot of these, we all know this. Once we get old, we all understand that there's like a cycle where something new comes along, like constitutive activity. Let's take an old one. As scientists, we get super excited about it. And so there's this upward curve where there's a lot of energy and a lot of really interesting positive results. And then we begin to see that the system isn't quite as simple as we thought it was, and that there are wrinkles, and so we go downhill, where we're like, and if we're not careful, we can throw the baby out with the bathwater. And then we finally reach that plateau where we understand what's going on. And I like to think of these new dimensions of GPCR research that we've gotten into as they're tools in a toolbox that help us solve problems. So the kinetics example for CRF was one where, if we hadn't have done the work necessary, 50 years ago to understand how drugs bind to receptors, if we hadn't done the radioligand binding assays and researched the kinetics, we would not have thought to look at that. So at that point, it became a tool in a toolbox where we say, wow, this is this now explains, and now we can apply it to make a better drug. So I think that's one very specific example, but I think we're going to see the same thing with biased agonism, where it's not as simple as we thought it was. But there's going to be somebody in a drug company, it's probably happened already, where they're working on a target. Drug A works, drug B doesn't. Drug A is biased, drug B isn't. And then they're going to be able to apply that to make the better drug. So I think rather than these concepts being sort of cathedrals that we have to engage in, I see them more as tools in a toolbox that we can use to solve problems, and they can be make or break. And we have plenty of examples of this where they've been just that. And Terry, if you go to Terry's presentations, he'll give you loads of examples of exactly how this is played out in real drug discovery programs, and it's great.
Exactly. That was his. That was a course I attended last year, which I thought was really fascinating. Where he made that connection between receptor function and why that compound, particularly, or that group of compounds, was a success in drug discovery, and why it was or it wasn't.
Yeah.
And then you could really go down to the molecular level and say, well, obviously, if we had checked this at a molecular level, we would have known that, it may or it may not work.
Yeah.
But then again, I think I think in industry things go by wave, and sometimes you have to ride the wave, and sometimes you have to try and convince leadership and say, no, actually look at the data. This is really a good way of going at it.
Yeah, absolutely.
And if the data supports your hypothesis or your idea, And at the end of the day, there might be a chance of succeeding. You just have to keep at it.
Yep, people will get behind you. That's great.
Definitely, and spend $500,000 in 18 months on animal studies.
Yes.
which it may sound like so much money, but it isn't. Again, it's a drop in the bucket.
It's crazy, isn't it? It's difficult to wrap your head around, but
it is. And when you think about it, how many hours or how many years do you need to work and end up with that amount in the bank?
Absolutely.
And some companies, just because it makes sense, because the data is supported and because they have to do the testing, just go ahead and pay that amount.
Yeah.
To get that data back. So it's just the proportions and the amount of money that goes into drug discovery is just unimaginable until you stop, think about it, and then you maybe work in industry, and that's when you realize, well, actually, $5,000 is nothing.
Yeah, it's the scale of the industry is difficult to wrap your head around. I mean,
it is.
Well, I used to work with somebody who did the large scale synthesis for drugs. And, and he made the point that if you add up the amount of aspirin that is taken in the world every day, it's many, many, many tons of aspirin taken every day. And he was mentioning a chemical synthesis he was involved in, where one of the starting points, they needed so much of it, it used to arrive in a rail car. And they would have a rail tanker, filled with 100 tons of this stuff, and they would just pipe it into and the scale is just mind-boggling.
Yeah, agreed. I think we don't think enough about these things. That little pill that you get out of a bottle, whether it's aspirin or ibuprofen, it's just made in these gigantic amounts, and then you have a machine that just spits them out at such a huge speed. There is this on the Discovery Channel, this Canadian, it's very short, I think 20 minutes, and it's called How It's Made. And it talks a lot about, there are a lot of videos about all of the things that made in an industrial setting. It can be donuts, it can be anything you can think of that needs to be made on a machine. And I enjoy watching it because you learn a lot of very useful things. Sure. And for example, even like specialty cars like Rolls-Royce and things like that. And they walk you through all these processes and you learn so much. And then you realize, well, actually to make a drug like aspirin, you must have that infrastructure as well.
Yeah, it's true.
To get it out. So the proportions are completely different, and just, trying to get to understand how these bigger things work is really revealing.
Yeah, absolutely.
So let's get back before we kind of wrap up to being an independent consultant. Mm-hmm. Let's say I'm a fresh PhD or I'm a fresh postdoc, and I know that academia is not for me. Industry, I'm not sure if I'm interested in. I want to be a consultant. What should I do?
So this first thing is to sort of recognize your talent. And I think newly minted PhDs kind of aren't really aware. I look back at myself at that age and I knew more about the literature then than I do now. I knew every paper on that topic.
Yeah.
You're one of only a handful of people that know what you know. And what you know is going to be more valuable, much more valuable than you think it is. That's the sort of overall idea. You'll need to do there are like two different types of consultant, right? There's the sort of old fossil consultant, which I guess is what I am. And then there's the consultant who knows how to do something that is really marketable. So I think for most of us working in GPCRs, they're still very, very exciting targets. So yeah. a lot of what we learn is very, very marketable. The two things I think you need are like the two things you need in any business. Number one is marketing. Now I'm not good at marketing, actually. It's really not my strong suit, as you know, Yamina. And so I'd strongly recommend doing a marketing class and learning how to do it. You might think you know how to do it, But there's tons of really obvious stuff that you'll learn just from taking a basic marketing class. It's really going to help you out. And my sister-in-law Amanda is in marketing, and she's basically done that job for me. So I've been really lucky. The second thing is, and this is more difficult when you're an early career researcher, is a network. So an awful lot of, I'm an old fossil consultant, so a lot of my stuff has come through the network that I built up over the years and through people who know me. That's tougher if you're new. So the marketing is more important, but you do know people. You're going to know a lot of people in your field. You're going to know people in industry. It's just a question of getting out there, broadening your network, and talking to people, just like you would with any other business. It's really interesting that you talk about this, Yamina. Until you raised it, I never thought about an early career researcher being a consultant, but I've really liked the idea. And it's just like you were saying. It's like when you're early career, you really, really know your topic, and we're living in a hyper specialized world now, where where somebody in a company working on a GPCR that you've worked on for the last 6 years, you know everything about that target. You know everything. A company decides that they want to work on that target. You might well be that person that can help them get that project off the ground. You're going to know basics about how to run an assay that company doesn't know. It's like, oh, you're going to need to add calcium in the buffer to get that assay to work. That they're probably not going to know. So, I'm not quite sure how this would work. This is a new idea. I think early career research is getting much more involved in consulting, but I think it's very, very interesting. And I could see myself working with an early career researcher that's really good at some big data questions. So like with kinetics, for example, where I'm working with Montana Molecular, and we're generating this data set where we're recording data every 2 seconds for 30 minutes on a 384 well plate. And that is a 50 megabyte file. So for you and me, that's like, how the heck are we going to do with this?
Yeah.
There's probably a 25-year-old postdoc sitting out there that deals with this every day that can say, I can analyze, you're taking 5 hours to analyze that data, I can do it for you in 20 minutes.
Exactly. I think it's a lot about, as you mentioned, recognizing the talent that you have and kind of understanding that. So again, thinking about it from my own perspective is that a lot of times the things that I find very easy to do, and you were talking about analyzing using the data, using the operational model, it seems easy for you because you're an expert at it, but it's not so trivial for a lot of us in the field. So I think you need to recognize what you're good at and really be good at it, and then leverage that and present it in a way that people can very easily understand that you're the expert.
Yeah. What do you think, Yamina? How would you do this if you were?
It's a tough one. So I would, it's interesting that you bring this up, but I think it's the right time to do this. So the way I see it, and one way of me, building relationships and putting myself out there is this very podcast.
Yeah.
And it's the newsletter, and it's my contribution to the GPCR field, where I get to meet people who either have companies or who either work in big pharma or consultants like you are. But at the end of the day, this is the network and the visibility that you need to build for yourself in order to get into consulting, I think. Because people know you. When somebody comes to you who, I don't know, has a Nobel Prize and says, hey, I love your podcast, can I come and talk to you?
Yeah.
That is just, there is, at the end of the day, there is no financial, because so let's take it a step back. Everybody needs money to live. That's one thing. But at the end of the day, when these opportunities come up to talk to so many interesting people, it's priceless.
Yep.
It's just priceless. So going into consulting, which is something I'm aspiring to do, the podcast, the relationship building, the summit is in one way to get to know people, get people to know each other, to connect with each other.
Yeah.
And also serve as a focal point. So let's say, Sam, you're looking for some data, and I can tell you, oh, I have the data, or oh, you should talk to this person who has the data, which we've done. I think that's the most rewarding thing ever.
Yeah, I agree.
And as a young PhD or as a young postdoc, you might think, well, I don't know anyone. No, that's not true. You know your supervisor.
Yeah.
You have people on your thesis committee who are established scientists, so you know them.
Yeah.
And the field, I think in every other field, it's small enough that you can say, well, look, this is X. I worked in X and I'm interested in this. Do you have 5 minutes?
Yeah. Yeah.
And that can make it or break it. And sometimes you get along very well with people. Sometimes you don't. But that's fine, because that's how you develop emotional intelligence, which you brought up earlier in this discussion. And that's how also you realize what type of people you want to work with.
Yeah. Yeah.
Because ideally, at least in my world, I want to work with people with whom I enjoy working, with whom we're collaborating, we're exchanging. And, we can have the biggest fight about the data because we don't agree, and then go grab a drink.
Yeah.
Like nothing happened before. And I see this, and the thought process that comes into mind is these movies where you have these lawyers and the movie, and they're against each other and they're fighting in court, and at the end of the day, hey, do you want to grab a drink? And yes, let's go. And then that's the type of relationship that I'm looking for.
Yeah, absolutely. Yeah.
So that's my way of seeing the whole field of consultancy. And as I mentioned, I listened to this podcast about consultants, and they were mentioning that there's 2 types of consultants.
Yep.
So one type where you come in and as you do, and you help with data analysis, and then you actually run through the process where the company or the customer generated data, and you try and solve their problem. That's one type of consulting. The other one, which is also called consulting, but it's kind of an odd thing, where you actually go into the company, and you full-time dedicate 5, 6, 7 months or a year with them and work as part of their team.
Yeah.
Which again is dependent on what you like and what you want to do.
Yeah. Yeah.
It's 2 different models I can see.
Yeah, that makes sense.
And you have to find what you like.
Yeah, it sounds to me like you mean, like, for you, a lot of it is connection. It's about connection. And I know, you do an amazing job with this, and it's you're kind of showing us how to do it, which is really nice.
Thank you for that. And it took me such a long time to figure out what I wanted to do. I didn't spend, the past 10 or 15 years thinking about, oh my God, what am I going to do? I really had fun, doing my master's, PhD, and postdoc, but there were times when I was wondering, am I in the right place? Am I doing the thing that, utilizes my full potential?
Yeah.
I know that I would not, and you mentioned this when you took the grant writing course, I would have not enjoyed being in academia.
Yeah.
Because writing grants is just mind-bogglingly difficult for me. I mean, even the papers that I've published, it was like pulling teeth. It was difficult, and not because I didn't know how to analyze the data. I just had such a hard time communicating or putting on paper what was in my head. Yeah. And there are other people, and I think the one person, and Michelle is really good at this, but Tom Sackmar he's just phenomenal at this. It's just incredible. And I'm like, how do you do it? And it's funny because I had this thought. So we in Tom's lab, we have or they have a visiting professor, Dr. Tom Haynes. I don't know if you've known him, but he's wonderful. He's 80 plus. He still runs 5 miles in the morning in Central Park, and he wrote his memoirs. And Dr. Haynes has a wonderful story. So if anyone wants to read his story, just go on Amazon and you can find it. And so, and then anyway, long story short, I was reading his book one day going to work, and I was reading the preface, and I was like, oh my God, how can you write? Who is the wonderful writer who writes such a beautiful preface to the book? And it turned out to be Tom. And, it was on the phone, so I didn't scroll ahead, and I was reading it and I had tears in my eyes. I was like, wow, such a beautiful wording. And then at the end I said, well, Tom Sackmeyer. I said, of course it's Tom. And some of us have that talent, and some of us, I don't have that talent. I mean, even writing an email sometimes, especially when it's complicated about data, I'd rather pick up the phone or go on Zoom, because it's just, I need my hands and I need to show and I need to point. So I think it's just, you need to find what you're really good at. And it turns out I enjoy talking to people.
Yeah.
And that's what makes my day.
You're really good at it, Yamina. You bring a lot of joy to the world with your talking and with your podcasts and the whole.
Thank you. Thank you. Yeah, we're always looking to talk to people. I've started interviewing PhD students and postdocs. I think we talk about different topics, obviously, but I think it's a great addition to the lineup of speakers we've had so far to the on the podcast.
Cool.
So 2 last questions. We talked a little bit about your aha moments as a scientist. Top 3. What would those be?
This is super personal, so it's hard to make this relatable, but I'll just tell it like it is. The first one I was doing my PhD, and I developed the math to explain the binding model that was in my head, loaded the math into Prism, and the line went right through all the middle of the data points. That was, I think people who do data analysis know what that high feels like. With it. The next one was the team aha moment, which was on CRF, where we measured the dissociation rates of the compounds. That was thrilling. Seeing Dimitri communicate that story was one of, a real joy. Then the third one was really the signaling kinetic stuff, realizing that we could analyze the data and that it's much, much simpler than we think it is. And that we can, no matter what the curve shape, we can measure the initial rate of signaling using a math trick. That was fairly exquisite. But these are very niche experiences of a pharmacology data analyst, but they were thrills. It's what make, I think of them in maybe not every day, but most days, and it helps keep me motivated. So I think it's a great question that you asked this, and I've heard a lot of responses on the podcast, and it's like, yeah, I know what that feels like. That was good.
I agree. And now that you were mentioning your top 3 aha moments, I think we should put together an article of compilation of the top 3 aha moments for all these guests, and see who, and try and find the number 1 of the top 3 through all the guests, because I think they're very personal, but I think since we're all in the same field, and a lot of us are molecular pharmacologists who just get tears in our eyes when you fit the curve and it's just perfect. And then you're like, even if I wanted to come up with the numbers, I could not have been able to do this. And look at this, how beautiful it is. I think we can all relate to this, as you mentioned.
Yeah. But these days it's, as I'm not doing research so much anymore, the real thrill these days is getting an email from a young scientist saying, I saw your paper, it was really interesting, I applied it to my data, and here's what we got. That is like, I think Brian Roth talked about this.
Yeah.
Talking to young scientists when they bring you their data it's just fantastic.
Yeah, I think you get to a point where you don't have to run the assays anymore. I find because it's difficult. It's an art. I mean, to me, maybe some of the people listening will think, well, what does she talk about? But to me, it's an art to run these assays, especially these live cell assays.
Yeah.
And then it's a lot of effort, it's a lot of work, and you get better at it over time as you get to work more with one specific GPCR or a family of GPCRs. But then when the data is just beautiful and you actually make sense of it, it's just that dopamine high that's just it's not comparable. I think to some extent, all of us scientists are kind of drug addicts, dopamine addicts, where we're chasing that, and it's not only the end results, but also the journey that took you there.
Yeah, you're right. You're right.
So where can interested parties find you, whether they're potential customers or whether they're students with beautiful data they analyzed?
So the email is the best bet for me. So sam.hoare@pharmacanics.com. I'm guessing there'll be a transcript where people.
There will be, yes.
And then, so email's the best. Then my website, I keep a website going, like a lot of people, which is www.pharmacanics.com. There's lots of free stuff on there. So data analysis tools like Prism templates and Excel simulators that you can play around with. So if you kind of want to get a feel for what I can offer you, then that's the place to go. Mm-hmm. I'm also on Twitter @PharmaChanic, which you can find too. But yeah, email's the best, and I'm always delighted to hear from anybody. So I live in upstate New York, out in the countryside, so it gets a bit lonely sometimes.
Yes, I was just going to mention that. So you started out in the UK and you went completely west here on the West Coast in the US, and then you just shifted a little bit back to the east here.
Yeah, it's nice, but it's always great to hear from people. And I should mention, actually, I guess this is an important point. I do my consulting, but then I do my research side gigs. So I don't charge any money to collaborate with people. So like, like there's Shivani and I are working together on some stuff, and she was like, "Oh, do I have to pay you to do this?" And if it's for that's pure collaboration. So this so there is a separation of church and state in what I do. So.
Yeah, I think it's wonderful that your side gig is actually the fun closeness, keeping that closeness to the basic research. Absolutely. And getting access to that phenomenal data. The other thing I wanted to mention is that so your talk at the Dr. GPCR Summit is still available. You can go look at it on YouTube. But also on our Dr. GPCR page, if you go and visit drgpcr.com/summit, then you'll get access to the program and you can access Sam's talk as well. And there is a link where you can access all sorts of valuable files, whether they're Prism templates or Excel templates. And Sam, you update it regularly.
Yeah.
So that if people need to take a look at that, they can definitely use it from there.
That was really useful, Yamina. Thank you. A lot of people are very grateful to you for hosting that. It's very helpful. Thank you.
Thank you. I think it goes back to collaborating. So my husband and I talk a lot about, how to spread the word on science, but also how to contribute to the field, or, how to solve big problems, including in the GPCR field, but other problems as well. And the summit idea was his idea.
Oh, fantastic.
And I said, well, wait a minute, let me just take the dog on a walk, think about it. And then I came back and I said, you know what, let's just do it. It's going to be a free resource. And, if we crash and burn, well, we have tried it.
Yeah.
And if it's triumph, then we'll take it as well. And as long as people find it useful, I found it amazing. And one point I'd like to make when it comes to junior scientists, I think any projects that you initiate and then you go through, whether it works out great or not, you learn a tremendous amount of things. And these skills are called soft skills. But I think, you can say that you've organized a meeting or you coordinated. And I was in email contact with all of the speakers. So it's 30 scientists through this whole experience. And, it generated such great conversations. And that's kind of how I met you as well.
Yeah, it's been great,
And it's been just a great experience. So go out there, explore, and don't be afraid to fail.
Absolutely. That's the message.
Super. Thank you so much, Sam. It's a great discussion.
Thanks so much, Yamina. Really appreciate it. Thank you.
Thanks. Bye-bye. Thank you for joining us for this Dr. GPCR podcast episode. I'd like to thank our guest, as well as our team members, Attila Forrest, Shivani Sajdev, and Jin Chung. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcasts. Another way to support us is to share the podcast, the newsletter, and the virtual cafe with your colleagues. Email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.